thank you, first of all, for having me here and your hospitality. This has been great to see the Dolphin Island Sea Lab. It's very exciting to be here, especially because I'm from New York and it's still snowing there. So even though you're having bad weather, I love it. So this is wonderful. So I'm actually going to talk to you today about the last chapter of my dissertation that was never published. And I'm doing this because I think it's really cool, but I'm trying to gain some hype because if I get enough hype about this, and that means I have to go home and write it up and be done with it. So let's just go and dive in with this talk. Okay, maybe that's not working. We'll do this. Whoops. Here we go. Okay, so let me just start off introducing you to Foraminifera. These are the marine protists that I work with. So Foraminifera are single-celled protists. They're not animals, they're zooplankton. And they create calcium carbonate shells, or what we call tests. And there's two main flavors of Foraminifera. They're benthic Foraminifera. There are those that live on the bottom of the seafloor or attached to more hard ground reef structures. So that's like these beauties up here. And there's planktic Foraminifera. So leads live in the warm mixed layer of the surface ocean. And they also can become stratified along the thermocline, that transitional area in the ocean between the warm surface and then the deeper ocean where temperatures are more steady. So we find them today in the world ocean from high to low latitudes. And we also, they have a fossil record that goes back to about 175 million years. So planktic Foraminifera are passive floaters. They don't have fins. They're not like fish or anything like that. So they are at the mercy of ocean currents to disperse and move across the global ocean. So the figure on the bottom left of the screen here is just modern surface ocean circulations and gyre systems on our Earth today. So they're outlined in red and yellow and orange here. And Foraminifera only establish viable populations where they are happy. So, for example, if you put me in the woods of New York and you tell me to live, I'm going to be grumpy because I don't want to hunt squirrels and eat acorns or anything like that. But if you put me in my house with my cats and my dogs and my husband in my kitchen, I'm going to be very, very happy. So Foraminifera is similar. So what you're looking at now on this figure on the right side of the screen here, you're looking at these populations. So this first one is Polyneatina spectabilis. So this is an extinct species of planktic Foraminifera. Its fossil record is only found in the equatorial regions of the Pacific. So there was something about the equatorial Pacific where it was really happy living, but nowhere else across the world. Its descendant, Polyneatina obliquulaculata, occurs in tropical to subtropical waters across the global ocean today. So it has a more global distribution. So again, we're seeing in these planktic Foraminifera, they're only establishing populations where conditions are right. There's been several studies published that show that planktic Foraminifera, their occurrences are controlled by temperature. So this figure in the middle is from Isabel Fenton and others. She published in 2016. This is modern core tops drilled along the world ocean. And the darker, cooler colors here indicate a lower number of species, whereas the warmer colors indicate higher number of species. So this is a latitudinal diversity gradient with diversity being highest in the equatorial regions and then decreasing towards the poles. So you can even kind of see that these tropical versus temperate assemblages are very different. So notice with some of these tropical species, some of them look like popcorn, but some have these cool little appendages coming out of their chest. But then if you look at the temperate water species, they all kind of look the same. Well, not to me, but if you're not trained to look at them, they do. But they all have much more conservative morphologies, conservative test sizes and shapes. Okay, so because Foraminifera are controlled by temperatures of the world ocean, they will only establish populations where they are happy, and they are at the mercy of ocean currents to disperse. They are excellent to use for evolutionary studies in the open marine realm, and also for biogeographic analyses. What are the drivers of their evolution through time and the abiotic factors that promote that? So one of the coolest biogeographic patterns that planktic Foraminifera and other protists exhibit is called bipolarity. Now, bipolarity is when we have populations of an organism in either hemisphere and they're very happy, but they do not occur in the subtropical to tropical oceans for some reason or another. We also call this anti-tropicality because, again, they just don't like those tropical environments. So we have found this in the literature for bacteria, viruses, and other marine protists. And another species that does this are other protozoans like the radiolarians. So what you're looking at on this figure at the bottom right of the screen is from Steffen-Jans and others in their 2013 paper. And they have 3 species of radiolarians plotted here. And all of these little shapes and dots on the map are where they occur. So you might notice right off the bat, quite simply, that there are no dots in the tropical to subtropical regions. They exhibit this bipolar pattern, biogeographic pattern, or anti-tropicality. So in 2000, Kate Darling and her colleagues, they published a paper, and their research motivation was how common is bipolarity in planktic foraminiferal lineages today. So what they did was conduct genetic analyses of 3 living species of planktic foraminifera that they caught from the Arctic and the Antarctic, and that's that figure down there at the bottom. Okay, so they caught foraminifera that were alive off the coast of Greenland, and they also caught them live off the coast of Antarctica, kind of off the coast of South America right there. And then they conducted genetic analyses of 3 of these species, Globigerina beloides, Turborotolita quinqueloba, and Neogloboquadrina pachyderma. What they did was basically, in simple terms, create a family tree of who's most closely related to who. So not a family tree like a branching family tree, but do you all know about like that 23andMe things where you can do genetic analyses and find your yeah, it's like that. That's what they were doing. They were looking within a species to see which were most closely related to others. And this is their results. They found that within these species, so this is, I've kind of circled it here in red. This is a Arctic and Antarctic relative, so they're very common, very closely related. And quinqueloba, same thing. The Arctic and Antarctic populations were very genetically closely related, and the same thing for Neogloboquadrina pachyderma. What this indicated was that these populations had experienced transtropical dispersal, meaning that they had moved from the northern hemisphere into the southern hemisphere, and or the southern hemisphere into the northern hemisphere to exchange genetic material within about the last 200,000 years. These authors went on to further hypothesize that this genetic exchange between the Arctic and Antarctic populations had actually been going on since the Miocene, so about 16 to 18 million years ago. So this begged a question: how did polar planktic foraminifera that do not like these tropical oceans and warmer waters, how do they disperse across tropical oceans? So Darling and others in their paper, and Norris and DeVargas in their follow-up 2000 paper, put forth 2 hypotheses. The first is that these species were using cooler water upwelling zones along coastal regions as stepping stones to move from one hemisphere into another. So here's beloides, and I've kind of mapped it on here. This is a NOAA map. All of the red areas on that map are where we have coastal upwelling, so cooler waters from the deeper ocean being pulled to the surface ocean, and also with high nutrients. So kind of using them as stepping stones. And if this did happen, did they occur during cooler periods in Earth's climate, such as during glacial and interglacial periods, or other times of ice expansion? So the goals of the study were pretty straightforward. We wanted to test their hypotheses. Do planktic foraminifera that exhibit a bipolar distribution, do they use these upwelling zones to disperse across tropical regions? And if they do, did that occur during times of cooler climates in Earth's history? Okay, so the approach that we took to do this was use the fossil record of the planktic foraminiferal genus called Globocanella. I'll tell you more about them in a minute. And we wanted to reconstruct their dispersal history through the Neogene and Quaternary periods. So let's talk about the geologic history first. So on the left side of the screen, what you're looking at is the geologic timescale from about 66 million years ago till today at the top. And in the center of this screen, you're seeing the benthic foraminiferal stable isotopic stack. So if you're not familiar with this, what we do is use the chemical signatures from benthic foraminifera, those that live on the bottom of the seafloor, and we obtain these from deep sea sediment cores, they're fossil material. And if that line trends towards the left, that means the Earth was cooler and had more ice. If it trends towards the right, that means the Earth was warmer and had less ice. So this is a good proxy for temperature and ice volume through Earth's history. So I've circled with this red box, put a red box around the Neogene. And essentially what this was from 23 to about 2.58 million years was a time of Antarctic ice growth. So we see this general overall cooling trend occurring during this time. And this cooling trend was punctuated by a few warming events. Now, if we move into the Quaternary, so this is about 2.58 million years ago till about today, we get the establishment of these really nice glacial and interglacial cycles when the Earth kind of went into this cycle oscillating between ice at the poles, but that ice grew and contracted through time. Essentially, what we're seeing during the entire Neogene and Quaternary periods, the time interval of this study, is our modern oceanic system, or our oceanic system coming into its like modern day configuration. We have modern day surface ocean circulation developing, as well as deep ocean circulation as well. Okay, now what I'm showing you on the left side of the figure is geologic time. Now we're zooming in until the about last 22 million years of Earth's history. So really focusing in on the Neogene and Quaternary. What I've plotted beside that is the benthic foraminiferal stable oxygen isotopic stack. Again, where it goes cooler and warmer is plotted at the top. And I've also, in all of these little colored boxes over top of that, indicated major climate and tectonic events through time. And we will not talk about all of them. We'll be here all day. I just want to point out two to you today. The first one that we're looking at is the Miocene climate optimum. So this was a time from about 17 to 14.7 million years ago. It is a time when atmospheric CO2 reached about 500 parts per million. So it's an analog warm period. It's a period of time that we like to study to see where we're going in the future. And Earth warmed up because we had eruption of the Columbia River basalts during this time. The warming led to, the increased atmospheric CO2 led to surface ocean temperatures that were 3 to 8 degrees Celsius warmer than today. The second event that happened right after that was the mid-Miocene climate transition. This was a time that occurred from about 14.2 to 13.8 million years ago. We had expansion of Antarctic ice sheets that occurred during this time, so our Earth is really cooling down. And that led the Southwest Pacific Ocean to cool down by about 6 to 7 degrees Celsius. And throughout this entire rest of the time period, what we see in the geochemical records is continued cooling, continued ice growth, but also increased equator to temperature, oh my gosh, I'm going tongue-tied here. Increased equator to pole temperature gradients also developing. So again, modern configuration occurring during this time. Okay, I mentioned that we're going to use the genus Globocanella. This is my favorite genus of plankton foraminifera. They are incredibly cute. It's okay if you disagree. I'll judge you a little bit. That's fine. But they inhabit thermocline waters today. There's only one living species, that's Globocanella inflata. And they really live only in the mid-latitude regions because they do prefer cooler water masses. Their fossil assemblages, these other 5 species that are up here, also have a largely anti-tropical or bipolar distribution. They have continuous evolving. They were continuously evolving through the study period. So what you're looking at now on the left side of the screen, you're just looking at geologic time at the bottom from 22 to 2 million years ago. And those gray bars represent the times at which these species were alive. The ones that are grayed out, we think these are just regional variants and they may have occurred only in the Southwest Pacific. That's something that we're still working on. But to summarize, this group is a model group to determine dispersal dynamics and investigating bipolarity and antitropicality through time. Okay, so what I did, I went into this database that we have called Neptune. And it's a database where we have all these plankton foraminiferal occurrences from deep sea sediment cores that have been taken through the years. I downloaded all 6 of those species occurrences from the database to find out where they were. And I ended up with 32 globally distributed sites. And that's what you're looking at on this map on the screen. This is a modern day sea surface temperature map. All of the black dots that are plotted on there are deep sea sediment sites that were drilled, and their numbers are right beside them. And in the gray lines, those are just modern day surface ocean currents and gyre systems. All of those sites were put on the same age model and time scale. That's what took the majority of time here. If you want to talk more about that, I'm happy to. But it was tedious, that's fine. And we specifically chose sites that lie in the high latitudes. So you can kind of see hole 609 way up here in the North Atlantic and the high West Pacific, and also in the tropics, and then also coming down here into the Southern Ocean. So there are places in the global ocean that are completely understudied and underdrilled compared to others. And that's one of the reasons why you see such an uneven distribution of sites across the world. Okay, it's not easy to talk about 32 sites. So what I did was split the ocean into 9 different regions so that I could talk about them more easily. So here's the names of those regions, and the colors of those names correspond to the circle color here. So for example, here's red, that's the Northwest Pacific, and that encompasses those 3 sites. And what I did after I had all that data is I plotted the first occurrences of each of those species at each of those sites. So this is a lot to look at. These are each of the first occurrences of each species. So when they first appear at each of the sites, and every symbol here represents a different region that I defined in the previous slide. So let's zoom into this a little bit more so I can explain it a little better. On the x-axis is latitude with the equator in the middle. On the y-axis is age in millions of years ago. So again, for example, you can see that this species first appeared at hole 806b in the equatorial Pacific right around, I can't see this that well, but around 16.5 million years ago. But there's two things I want to point out here. We did take error into consideration when we were plotting these things. So there are error bars here. Some of these sites have large error bars associated with the first occurrence of that species. So this one for 1123b, for example, has an error of almost 2 million years. That's okay. We did the best we could. That makes interpreting their dispersal history pretty tricky. The other thing that we noted was that there's high diacriticity for some regions of the world ocean. So all of these orange dots here, those all represent the Southwest Pacific Ocean. And notice that this species first occurs there anywhere from like 18.5 to 15.5 million years ago. And that's just in one region. So that also makes things pretty tricky. So simply what I did was take this data, doing the best that I could, trace where they first evolved. So in this case, this one evolved in the South Atlantic, and trace their dispersal pattern through time across the world ocean. I'm going to tell you some other things that I found that were pretty interesting. So what I'm showing you now on this slide, you've seen this data before. So this is just that geologic timescale, the benthic foraminiferal stack with all those events here. These are the symbols for those 9 regions that I defined. And this is a phylogeny or family tree of all 6 of those species. I did not come up with this one. This was already published. And then what I did was plot all of this data right on top of this tree, these ranges of the species, to see what happened and what patterns fell out. So there's a few things I want to point out. First of all new species that evolved first evolved in the southern hemisphere, which was really neat to see. Nobody evolves in the northern hemisphere first. And we get a lot of new species in the early Miocene. And then after that global warming event, the Miocene climate optimum. The third thing that I want to point out is that most of the long distance dispersal, the movement of these species across the global ocean, occurs after global cooling, after we get past the Miocene climate optimum. Okay. I mentioned that I traced out these species dispersal patterns across the global ocean, and that's what I'm showing you in these 4 maps. So these are from 4 species. Simply, these are just modern day sea surface temperature maps like I've showed you previously. The dark black line indicates the dispersal pathway of each species, and each white dot on here represents the site at which it occurs at, and there's a small number beside that dot that represents the age in millions of years, the timing of which that species arrives at that site. It's small and unreadable. It doesn't matter. But I want to point out some trends here that I found that I think are pretty cool. The first thing is, so remember I mentioned that all species evolve first in the southern hemisphere. They use the Antarctic circumpolar current, this major current that whips around Antarctica. They use it like a carousel. So fun fact, Binghamton, New York is the carousel capital of the world. So I would be kind of a bad researcher if I didn't say, mention a carousel to show a carousel in this talk. But they're using the ACC as a carousel. So they're evolving in one ocean basin in the southern hemisphere, jumping on the ACC on that carousel, and they're using that as a means to disperse into another ocean basin. After they arrive into a new ocean basin, they are then using these eastern boundary currents. Those are outlined by the pink boxes here. They are generally cooler water currents. They occur on the eastern side of ocean basins associated with gyre systems. And they're also moving into the northern hemisphere using western boundary currents. They are more warm water currents that occur on the western side of these ocean basins and gyre systems. Okay, but now I want to get to the meat and potatoes of that question that I wanted to ask in the first place. Do plankton foraminifera that exhibit a bipolar distribution use upwelling zones to disperse across the global ocean, and especially these tropical regions? And if they do, does that occur during cooler periods in Earth's history? So to answer this question, we're going to laser focus in on Globocanella praescicula's dispersal pattern, as well as that as Globocanella myosea. Okay, just want to explain this slide to you really quickly before we get going here. You're looking at the last 19 million years of Earth's history on the geologic timescale on the left. Again, all of our events are plotted here with the benthic foraminiferal oxygen isotope stack, showing you when Earth got cooler and got warmer. I am showing you a modern day sea surface temperature map, and the gray lines again are modern day surface ocean circulation and the major currents, the sites at which this species occurs at and the timing at which it does. And I'm going to walk you through its dispersal pattern. So the timing at which will show up here. So you see a number 1 with an arrow here. That's where it first evolves. I will show you its dispersal pathway on the map. I will tell it to you in the words. So let's get started. Okay, so Globocanella praescutula, it evolves in the Southwest Pacific Ocean somewhere between 18.5 and 20 million years ago. After that, it disperses into the eastern equatorial Pacific Ocean at around 14.1 million years ago. And look when this occurs. It's during the Miocene climate transition when Earth cooled down. From geochemical studies conducted by Holborn and others in 2014, they found that during this time, there was increased equatorial upwelling that occurred in this region due to global cooling. So let me just explain that really quickly. So here's another modern sea surface temperature map on the left. We're zooming in into the eastern equatorial Pacific. So notice, first of all, that this is much cooler water than the surrounding equatorial Pacific. That's because there are winds that are blowing from the east to the west, pushing the warm water away into the western equatorial Pacific and pulling cooler thermocline waters to the surface. Okay, so this causes cooler waters to occur off the coast of South America. It also has high chlorophyll concentrations because these deep waters are really rich in nutrients. Okay, so this whole system that's going on today started to kick up and really intensify during the Miocene climate transition when Antarctic ice started to grow and the Earth started to cool down. After that, that species then appears in the Northwest Pacific at 13.39 million years, right after the mid-Miocene climate transition. So the dispersal pathways and biogeographic data from this species indicate that, yes, it actually did use equatorial upwelling zones as a stepping stone to cross the tropical ocean and move into the northern hemisphere. So now we're going to look at Myosia, same setup here. I'm going to walk you through its dispersal pattern. So this species first evolves in the South Atlantic at around 18.5 million years ago. It then dispersed into the Southwest Pacific Ocean by 17.7, so using the ACC to then migrate into a new ocean basin. It then disperses into the Western Equatorial Pacific Ocean around 16.3 and hangs out there for about almost 3 million years, not quite 3 million years. So the occurrences of this species in the fossil record at the site are super, super rare. So my PhD advisor actually worked on this site, named Site 806, in the Western Equatorial Pacific. So that data comes from him and his first PhD student. And he told me, Adriane, the occurrences of this species there are rare as pin's teeth. And I was like, cool. So that indicated to us that this species was trying to disperse across the tropical ocean. But remember how I said the populations, that it's not going to establish a robust population if the conditions aren't right. The conditions seem to not be right for it. It was able to reproduce maybe a little bit just to make it into the fossil record, but that was about it. However, at 13.78 million years, notice in the mid-Miocene climate transition again, that species is then able to make it into the Northwest Pacific Ocean. Another geochemical study that was published by Gasparini and Kennett in 1993 found that there was a cooling of the thermocline in the Western Equatorial Pacific, again due to cooling of the Earth due to ice expansion on Antarctica. So let's talk about that for a second. So now what I'm showing you is this box plot across the the Equatorial Pacific. Here's the western part. That purple arrow represents where that site is that I just told you about. We just talked about upwelling on the Western Equatorial Pacific and all of that piling up of warm water in the Eastern Equatorial Pacific, piling up in the Western Equatorial Pacific. Okay, so the geochemical data from Gasparini and Kennett, let me just walk you through this as well. So they used the geochemical data from the benthic foraminifera. These are bottom-dwelling foraminifera, so they record a bottom water signal and temperature change. And they also looked at plankton foraminifera, their temperature signal or oxygen 18 data that live in the very warm surface ocean. So they were looking at the gradient between these two. And right around the time the species dispersed into the Northwest Pacific is the time we find the water masses differentiating. So the bottom waters got cooler, the surface got a little warmer. They also played the same game with bottom-dwelling benthic foraminifera and those that live in the deep thermocline, that transitional area in the upper ocean. And they also found a large gradient that was developing. So in other words, what they're seeing is cooling of the bottom waters due to Antarctic ice, forming cooler bottom waters, and also warming surface waters and the differentiation of this warm surface layer, a cooling thermocline, and even cooler bottom waters. Okay, so the biogeographic pattern from this species, Globocanella myosia, suggests cooling thermoclines were also used by species as an additional strategy to cross the equatorial regions. All right. This actually, I'm going to switch gears kind of abruptly for a minute, but I'm going to tie this all together. So my job on the ship as a IODP scientist is a biostratigrapher, and I use the plankton I was just telling you about to tell the age of rocks and sediments that we pull up. So biostratigraphy, bio equals life. Stratigraphy is a branch of geology that deals with putting time and age into the rock and sediment record. So I'm using those tiny fossils to tell the age of sediments, and we use what's called a biozonation scheme to do this. So when you're drilling sediments in the ocean, you have to go pretty quickly, so you can't get an exact age. But what we come up with using the first and last occurrences of key species of thoriums are what we call biozones. So these first and last occurrences of species may differ across the ocean basin. So biostratigraphy really relies on understanding biogeographic patterns of these fossil plankton that we use, and also detailed knowledge of when species may appear or disappear at certain sites through time. So today, when we're on the ship doing this kind of stuff, there is a global scheme, biozonation scheme, that we use to refer back to tell the age of these sediments. So this is constructed for sites that lie in the subtropical to tropical water masses today. And it's really robustly used. It's been updated, and you're seeing just a part of it on the screen. But the issue with this is that not all of our ocean is tropical to subtropical, right? We've just learned and heard about that there are some ocean or some water masses that are very polar to subpolar, and then we have the temperate water masses in the mid-latitude. So that zonation scheme actually isn't appropriate for everywhere in the ocean. Going back to the research and the patterns that I showed you reconstructed from the global canelids, we know that species appeared in the northern hemisphere much later than they did in the south hemisphere, and that because they evolved in the southern hemisphere first, and then later migrated up into the northern hemisphere. So the question now remains, what implications does this have for biostratigraphy, and assuming that these species may occur everywhere across the global ocean at the same time? So this is another little study I did as part of my postdoc. I call it my pandemic project because I was supposed to be doing geochemistry for my postdoc research, but I was at home, so I got my husband to actually help me do research. It was actually kind of cool. He's on a paper, actually on this paper that we published together. It's great. So what we did, we looked at the sites that lied across the Western Pacific Ocean, and we wanted to know what were the differences in species first and last occurrences at these sites. We wanted to quantify that. So these sites span from the mid-latitudes all the way to the tropics in the northern and southern hemisphere, namely the Croceo Current extension in the northern hemisphere, this whopping western boundary current, and the Tasman front that's down here, and the East Australian current and extension. Okay, so we wanted to compare the timing of the species first and last occurrences between that tropical zonation scheme that I showed you, this, and for those that I developed and calibrated from the site that from the study that I just told you about. So we used 31 species, and we used a total of 43 first and last occurrence events of these different species to look at the differences in timings. So we mapped out their first and last occurrences through time across the Western Pacific, again, to get an idea of how their dispersal patterns affect our ability to tell the age of sediments through time. I'm just going to show you some of this data pretty quickly. What you're looking at are some species first occurrences. Here we are, the geologic time scale again. On the left, this goes down to 12 million years ago. Each of these gray columns represent a site in our study across the Western Pacific. The one in the center that says tropical, those are the dates of these species from that biozonation scheme I told you about in the very beginning. That one, that's probably not good to use everywhere. So what we did is just simply plot on those graphs when these species first appeared or first evolved at these sites through time. So there's something I want to point out. Some species first appear in the Northwest Pacific. So like Globogeronella callida, here's callida, it's very cute. And then they later appear in the tropics. And then they, lastly, in the Southwest Pacific Ocean. Some species first appear in the tropics. So if we look at Polyneatina primalis down here, it first appears in the tropical oceans, and then it later spreads much later into the mid-latitudes. You see that kind of U-shaped pattern that's going on here. Come on, clicker. Oh, I'm pushing the wrong button. Okay. More species first occurrences. So for the globocannelids, here's Inflata, Puncticulata, Conomysia. No surprise here, they first occur in the Southwest Pacific Ocean. That's where they first evolved. They then later migrate into the tropics, and then later even more into the Northwest Pacific Ocean. So there's a few of these. If you trace the purple lines, they kind of step up into the Northern Hemisphere. If we look at the last occurrences of some of these species, Globogerinoides and Poliniatina, so they're outlined here in those blue boxes. What they do, they first disappear from the Southwest Pacific, so we can kind of see this here, and then they later disappear from the tropics and then from the Northwest Pacific. So they kind of constrict their range back into the Northern Hemisphere, which is kind of an interesting pattern. Globoquadrina is really interesting. It's one of our main species that we use to tell us when we're in almost to the Pliocene. But I really want to point this out. This is amazing. In the tropics, that holds true. It marks the end of the Miocene. But if you get into the mid-latitudes, it doesn't indicate the end of the Miocene. It actually indicates more of the middle Miocene. So if you were in the mid-latitudes of the Southwest or Northwest Pacific Ocean and you found this species in your sediments, you would actually be wrong on your age by several million years. So that's kind of interesting. So we see two major dispersal types. We see this gradual dispersal from the Southwest Pacific into the Northwest Pacific, which is basically what was the first part of this talk. We see this migration pattern in species. The second migration pattern we see is a stepwise dispersal from the Southwest Pacific in purple, and they leap into the Northern Hemisphere up here. So we see that in a few different species as well. Okay, so the whole goal of this was to quantify the differences in these species' first and last occurrences across these different sites. So the average difference of species' first and last occurrences in the Northwest Pacific Ocean was 1.39 million years. In the Southwest Pacific Ocean, that's over 2 million years. And if we look at the Western Equatorial Pacific, these two sites that lie very closely to one another, the differences in these species when they first occur or were extinct in that area is 0.8 million years. This matters greatly because planktic foraminifera are used as first-order age control in marine sedimentary sections. So they are the first thing that we will go to get an estimate of the age of those sediments that we are drilling. So if we only use that tropical or global tropical to subtropical age model, we will get vastly different ages of our sediments, and it will be way off and very wrong. And scientists, when we're on the ships, actually use that age model to pick out samples in their core where they want to take samples and do further research. So if we're off by millions of years, the scientists may not be sampling even in the correct interval that they're interested in. So there's a few expeditions that are scheduled and sailing and that have already scaled. Expedition 400, these should be switched. Sailed last year. Expedition 403, I'm sailing on that this summer, will also be in the high latitudes. Expedition 402 is currently sailing in the Mediterranean Ocean. My PhD student Ravi is currently out there right now. Expedition 401 sailed end of last year. And Expedition 406 is going to be drilled off the New England coast sometime hopefully next year. So we really need more regionally constructed biostratigraphic schemes to use at these sites, especially in the mid to high latitudes. Otherwise, our sediments and the age of those sediments will be highly off. Okay, just to conclude and wrap up here. So Darling and others in their 2000 study and Norris and DeVargas in their 2000 paper hypothesized that species of planktic foraminifera used upwelling zones along coastal regions as stepping stones to move from one hemisphere into another and to get across tropical oceans. And this may have occurred during cooler periods in Earth's climate. So our first question, do they do that? Do species that exhibit a bipolar distribution use these upwelling zones to disperse across the tropics? We found that yes, they do. In addition, they also utilize cooling thermoclines as an additional strategy to move from one high latitude region into another. Do these events occur during cooler periods in Earth's history? And in fact, yeah, we did find that. Transequatorial dispersal of Globocanella myosia and Praescutula both occurred during the Miocene climate transition when Antarctic ice sheets grew and developed. And these dispersal events across hemispheres cause differences in these species' first and last occurrences that can really mess up our age interpretations if we're not calibrating their first and last occurrences at a regional scale. Okay, so just some acknowledgments. Of course, IODP made it possible for us travel around to different universities, museums, and labs. So I have to thank them for bringing me here today and all of you as well. Really appreciated it. This research was funded by the Cushman Foundation for Foraminiferal Research, namely their Joanna M. Riesing Foraminiferal or Fellowship that paid for the last year of my PhD. This is Joanna Riesing. She is barefoot on a bench in her garden holding a cat. So I feel like I would have gotten along great with her. So that's all I've got. I'm happy to take any questions. Thank you all.